My lab is a developmental neurobiology lab, which means that we really want to understand how brains are built. We know that the human brain or mammalian brains have very limited regenerative capacity. So if cells or if there's brain damage, the only way for us to fix that is to reintroduce cells that are not damaged or not subject to some disease process. My lab focuses on using the fruit fly called Drosophila melanogaster as a model, and we particularly focus on the visual system to study how the visual system forms as a model of nervous system development. We do a lot of imaging, so we use fancy microscopes to visualize where genes are expressed and where transcripts localize in cells, where proteins localize, how cells interact with each other and form connections with each other. We can watch some of these processes live, so we can watch cells moving through the brain during development. And in addition to all of the wet lab work, we also do a lot of bioinformatics. I think a major challenge that we face frequently, but is also possibly part of the fun of doing research, is having to become an expert in an area that you're not an expert in. And this can be in a research topic, but it can also be in learning new techniques. There are a lot of exciting projects in the lab. Maybe I'll tell you about not one project, but maybe a whole program of research that the majority of the lab right now is focused on, which is how a neuron starts to differentiate. And what I mean by that is, how do you go from an undifferentiated cell that's not really doing anything too exciting to the cell actually committing and locking in being a neuron? So can we reprogram skin cells or a cell in the leg of a fly or the wing of a fly, given this knowledge that we'll have uncovered, to make these cells that normally wouldn't become neurons? Can we artificially reprogram them to become neurons? So that, I think, will be the next exciting direction.